Hello everyone, welcome back to my channel. So in this video we are going to basically talk about the Watson and Crick model. But before moving ahead into that, let us just recall once again the history of the genetics, or from when the search of the genetic material began, or that. So firstly, there was the scientist called Mendel who first told about the factor. He was discovering the molecule or the thing which is carrying all the characters, which is passing all the characters from one generation to the next generation, and Mendel named that molecule as factor. Next, one more scientist came into the story, that was T.H. Morgan. T.H. Morgan said that opposed to one of the concept of Mendel, or one of the law of Mendel, that independence assortment, that all the time the factors do not pass into the next generation independently. Sometimes what happens, the factors can be passed to the next generation together. That event is considered as linkage. So that concept came from T.H. Morgan's story. Then two more scientists came who gave the chromosomal theory of inheritance. Those were Sutton and Boveri. So these were two scientists who gave the chromosomal theory of inheritance. Then came Friedrich Miescher. Friedrich Miescher was the scientist who first said about the nuclei. And then came Frederick Griffith. Frederick Griffith told us about the transforming principle. So till date, nobody knew that DNA is the genetic material. Everyone was searching for the genetic material, but everyone was giving them different names. Mendel gave factor, then Griffith gave transforming principle. Then to establish that what is the transforming principle, is that protein or DNA or RNA, to find it out, Avery, MacLeod and McCarthy did one experiment, the repeated Griffith's experiment, and they proved that it is nothing but DNA. And after that, Hershey and Chase came, who said about the genetic material, that is nothing but DNA. So Hershey and Chase were the scientists who proved that DNA is the genetic material, not the protein. So in this way, the discovery of the genetic material happened, or search for the genetic material, or urge to search for the genetic material happened. Now we are thinking about the structure, how the DNA is looking like, what the DNA is consisting of, that is still yet known. But what is the exact structure of the DNA, that was unknown to all the scientists. So in that scenario, two scientists or two names come in the history of genetics, that is Maurice Wilkins and Rosalind Franklin. Very important. Their all the activities and everything, whatever they did, Rosalind Franklin and Maurice Wilkins were the people who actually did one experiment. Basically, Rosalind Franklin did one experiment, which is, in today's world, it is known as X-ray crystallography technique, or X-ray diffraction technology. So X-ray diffraction technology, what it is actually, if you will see here, that she took in one test tube the DNA strand she took. She didn't know what is the structure, how does it look like, is it like a round shape, is it like square shape, or how it is. That was unknown to everybody. So she started doing this experiment. This was the source of the X-ray. These are the source of X-ray. And from here, X-rays were passed through these, and all the X-rays were diffracted, and these were falling into the plate. Here one picture plate was being kept so that the structure can be caught into the plate. So on the plate, she found that this kind of structure was there. this kind of structure was there. So she discovered this thing. This is the structure which is found by Rosalind Franklin. She was actually a scholar of who was working under Maurice Wilkins, and two more scientists later on, they also found out some of the factors like how the base pairing and what are the bonds and how are they situated, located, about everything they said. But the basic structure, that is the DNA double helical structure, what this structure is called, DNA double helical structure. This double helical structure of DNA was first discovered by Rosalind Franklin. But unfortunately, while doing this X-ray crystallography technique or X-ray diffraction technique, she got a cancer and she died for it. And that is why, unfortunately, her name was not nominated for Nobel Prize. And the Nobel Prize was given to this Watson, Crick and Maurice Wilkins. These three persons got this Nobel Prize. But actually the work, basic work, was done by Rosalind Franklin. Okay, so this was the story of the little bit of story kind of a thing, or which is very important and related to the history of the genetics. Now let us move ahead into to see that what is the structure, exactly structure of this. While doing all of these things, there was another more biochemist whose name was Erwin Chargaff. Erwin Chargaff was there, who said about the structure or chemical makeup of DNA. He said that I do not bother, I do not care that what is the basically physical structure of DNA. He said that I am a biochemist, so I have to deal with the chemical structure or chemical makeup of DNA. So I will definitely deal with the chemical makeup of DNA. So he dealt with it, and he said, he gave some of the rules or some of the laws. He said that he knew that DNA is consisting of sugar, base and phosphate. Now in the nitrogenous bases what are present in DNA, that was also known to him. So he said that adenine, all the purines will always form a bond with pyrimidine. Adenine will always find a bond with the thymine, and guanine will always form a bond with cytosine. This was his first law. Then Chargaff gave the second rule, that is the number of adenine. If adenine is forming the bond with thymine and guanine is forming bond with cytosine, so it is obvious that the number of adenine and number of thymine, that is number of purines and number of pyrimidines, will be similar. So that is why he said that adenine and guanine as well as thymine and cytosine, everything, number of them will be similar. And as well as they said that adenine and guanine divided by thymine plus cytosine will always, this ratio will be 1. So proportion of them will be same, and this will be 1. These were the rules which were given by Chargaff. So now let us come into this story, where the physical structure, the scientists are searching for the physical structure, and a double helical structure has already been discovered. So now Watson Crick saw that yes, this structure is exactly like a ladder which is running parallel to each other, and they are intertwined with each other. So let me first draw the ladder like structure here. And these are the steps of the ladder. We can consider these as the steps of the ladder, but some of the scientists say that these are the rungs of the ladder. These are the rungs of the ladder. And they also said that there will always be two opposite poles. One will be 5' to 3', and in another one there will be 3' to 5'. Okay, so this is the structure of the DNA. They said when this will be intertwined, then this kind of double helical structure will be formed. Now why always the purine, one purine will always form a bond with pyrimidine? Why this is true? Why always adenine will form a bond with thymine and guanine will always form a bond with cytosine? If you remember it or not, that I don't know, but in my previous video I have already said you that the adenine, guanine, these are consisting of 2 nitrogenous rings. But in case of thymine, cytosine, uracil, these are consisting of only one ring. So in this case, what is happening? Adenine is consisting of 2 rings and thymine is consisting of 1 ring. Guanine is consisting of 2 rings. Cytosine is consisting of 1 ring. So let us just consider that thymine is forming a bond with adenine and cytosine is forming a bond with guanine. So in all of these cases, you can see that they are forming a exactly similar kind of a structure, exactly straight line kind of a structure. But if supposedly these were not like this, adenine was forming a bond with guanine, cytosine was forming a bond with thymine, then adenine was forming a bond with thymine, then once again say for example guanine is forming a bond with adenine. So in all of these cases you can see there is no consistency. The structure is like this kind of a structure we used to get. But this is not true. The concept is this. The true concept about the structure of DNA is this. This will always be parallel to each other. The runs will always be parallel to each other. That is why this concept just finished. This was just diminished. This was a very wrong concept about the structure of the DNA. That is why the first point which came into Watson and Crick's model, that is the DNA double helical structure is always consisting of one end which is consisting of 5' end and another is always consisting of 3' end. They are intertwined with each other and the runs, these are running parallel to each other. The backbone, now let us move ahead into the basic structure, that is the backbone. This is the backbone, this is the backbone. So backbone is consisting of sugar and phosphate molecule. So if you remember the structure of the sugar and phosphate and base, so you saw that if supposedly this is the sugar, in one hand this was holding the phosphate, in other hand this was holding the nitrogenous base. I am writing it down like adenine, and with this the next sugar molecule comes into the story. This is also holding phosphate with it, and in another hand this is also holding the nitrogenous base in it. So how they will form a bond? Here comes the role of the bond which is forming between these two molecules. So in this way sugar, phosphate are present alternatively. So in this case phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. In this case also phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So that is why it is said that DNA backbone is made up of alternative sugar phosphate, or DNA is made up of alternative sugar phosphate backbone. This is the reason behind it. Why is it called as alternative sugar phosphate backbone? This is the reason behind this. So in this way these are forming bond between them, and as one sugar molecule is holding, in one way it is holding this phosphate, in another way this is holding another phosphate. There is the sugar, there is the another phosphate. These are the nitrogenous bases, adenine, guanine, another rest of the bases. So this is the bond by which these are linking. Two molecules are linking, two nucleotides are linking. So this linking or the bond between them, this is known as phosphodiester bond. As you can see phosphate, and it is present in both of the sides. That is why this is known as phosphodiester bond. So the bond between two nucleotides are called as phosphodiester bond. Okay, because in between them the phosphate is coming, and in both the sides there are the esterification happening. That's why the name of the bond is phosphodiester bond. And with the sugar, the nitrogenous bases are also linking. As you can see that with the sugar, the nitrogenous bases are also holding. So how will it hold it? It is holding it with the help of glycosidic bond. With the help of glycosidic bond. So glycosidic bond is present between the sugar and the nitrogenous base. Between the sugar and the nitrogenous base, the glycosidic bond is present. And with the complementary base pairing, there will be always complementary base pairing with each other. That means one purine molecule will always form a bond with one pyrimidine molecule. And in between them, there is the hydrogen bond, presence of hydrogen bond. There will be 2 hydrogen bond between adenine and thymine, and there will be 3 hydrogen bond between guanine and cytosine. These are nothing but the hydrogen bond. These are the hydrogen bond. So these are the bonds or chemical bond or link up between the two different types of chemicals or different types of molecules. I hope that this scenario is clear. Now once again let us move ahead into the Watson and Crick structure. So here comes the alternative sugar phase, sugar phosphate backbone, and all the sugar is holding always the bases, nitrogenous bases. So if it is holding A, from opposite side there will always come T. If it is holding G, then from opposite side there will always come C, and between them there will be the bond formation. So in this way it is forming a very compact kind of a structure, due to which it is very stable in nature. It is not at all that much reactive with any kind of chemical, that any sort of chemical will come, any sort of molecule will come and it will start reacting with it. No, that will never happen to it. That is why also the nitrogenous bases are inwardly present. These are not hanging outwardly. These are hanging always inwardly, so that there will be no reaction between the molecules. That is also one of the main reasons. So now if that will be twisted or that will be intertwined, then this will form this kind of a structure. This kind of a structure will be formed between them, and so they said that there is a major group and there is a minor group in one complete turn. If you will see that this is a major group and there is also a minor group, this is the minor group. 2 group will always be there in one complete turn. And they also say that in one complete turn there will be 10 base pairs present. So let me draw it here, like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So in this way 10 base pairs will always be present. And what will be the distance between them? Between one group, that will be 34 angstrom. Distance in one complete turn. This is consisting of a complete turn. And this turn is consisting of one major group, one minor group. And in one major, one minor, it is consisting of 10 base pairs. And the distance between one total turn to the next turn is 34 angstrom. That means within one base pair, what is the distance? As it is completely consisting of 10 base pairs and distance between them is this 34 angstrom. So each base pair will have obviously 34 divided by 10, that means 3.4 angstrom. So between 2 bases, if I'll see here like this, between 2 bases there will be the distance of 3.4 angstrom. And all of this scenario is always constant. There is no any other kind of concept or no any other kind of situation where this 34 angstrom will be changed, 3.4 angstrom will be changed, distance between them will be changed, 5 prime will come close to each other. No, never. That will never happen. All of these things are always constant in nature. And all of these are known as Watson and Crick model. This model was given by Watson and Crick because they saw that Chargaff also said this kind of thing, and they also saw that this kind of structure is coming, physical structure is coming from Rosalind Franklin's X-ray diffraction process. So they just did 2 + 2 4, and they draw this kind of a hypothesis of the total structure of the DNA. And from here the double helical structure of DNA and that concept came. So here I am ending my today's lecture. I hope that this video was helpful to you. If so, then please don't forget to like, share and subscribe to my channel, and please comment down below. Thank you.